Good morning. Thank you all for registering for today's webinar related to designing liquid-to-liquid extraction columns. I am Mauricio Villegas, and I am the Business Development Manager at Koch Modular. This will be the first webinar in a series. Our next webinar will be discussing the benefits of truckable module process systems, so stay tuned for a future announcement. During today's webinar, you will hear from our subject matter experts about the considerations when designing liquid-to-liquid extraction columns. For those unfamiliar with liquid-to-liquid extraction columns, it serves as an important tool for those separation processes which are otherwise difficult to perform using conventional techniques. Furthermore, our columns find their way into most markets such as specialty chemicals and performance materials, biofuels and biochemistry, pharmaceuticals, food and flavor, plastics and rare earth metal recycling, and other markets. So a little bit about Koch Modular Process Systems. Koch Modular has been designing and delivering modular process systems for over 25 years, and we are a JV partner with Koch-Glitch, one of the world's most prominent suppliers of mass transfer equipment and part of the Koch Industries family. Since our inception, all our process systems are delivered modularly, thus minimizing field work and resources. Whether pilot scale or commercial scale, Koch Modular can design and deliver modular process systems ranging from small scale systems with one single module to a large scale system comprised of dozens of modules, such as the system shown here in this picture. When it comes to liquid-to-liquid extraction, we are the industry leader, having acquired Koch-Glitch liquid-to-liquid extraction technology as part of our joint venture, thus bringing over 60 years of experience and having delivered over 300 liquid-to-liquid extraction column systems worldwide. As a result, we are the exclusive global provider of Koch-Glitch technology, thus allowing us to provide performance guarantees to all our systems. Today, our presenters are Don Glatz and Brendan Cross. Don Glatz is a manager of extraction technology at Koch Modular. Don brings 25 years of liquid-to-liquid extraction experience and has published numerous papers and articles covering this subject. Don's activities include the evaluation and optimization of extraction processes, plus scale-up and design extraction systems. Don holds a BS in Chemical Engineering from Rensselaer Polytechnic Institute and MBA from Fairleigh Dickinson University. Brendan Cross is a senior process engineer at Koch Modular that works in the extraction technology group. Brendan is responsible for liquid-to-liquid extraction application, evaluation, extraction column and pilot test design, and commissioning and process startup. Brendan brings over 10 years experience in distillation and process development. Brendan holds a BS in Chemical Engineering from Columbia University. So again, thank you all for joining. And before I hand it over to our presenters, I just want to mention that our chat line is open and we'll be holding all the questions towards the end, and we'll leave about 10 to 15 minutes to cover your answers. Thank you.
Good morning, all. This is Don Glatz talking, and thank you, Mauricio, for that kind introduction. I'll be doing the first part of the presentation, and then I'll be turning it over to Brendan for the second part of our presentation. So just starting out here this morning, first slide, we're showing typical places where liquid-liquid extraction is utilized. We're going to focus primarily on the top three because most of the work in liquid-liquid extraction that we get involved with involves these top three areas. And certainly the top two are similar. When you have either high boiling organics or non-volatile components in an aqueous stream, it's very, very expensive to try and distill all that water away from those components. So in those particular instances, often liquid-liquid extraction is a much more economical solution to removing those materials from the aqueous stream and then eventually recovering them. The third one, many times water is used as the solvent in an extraction process. Typically, if you have an organic stream with something to remove and that something can be is very miscible in water, a lot of people call this a water wash in their plants, but in actuality, they are in fact doing a liquid-liquid extraction process. Here's some industries that we get involved with. This is just some. We get involved with more than just these, and we're trying to show some very typical types of extraction processes that you'd often see. Certainly, we do a lot of work in the CPI area. We mentioned there acetic acid, a washing of acids and bases, and sometimes just recovery of very valuable materials. In metals and mining, there's a lot of different metals that can be extracted and recovered by a liquid-liquid extraction process. Many times in polymer processes, we get involved. Caprolactam is certainly a well-known process where 3 extraction columns are typically used, and there's probably 20 or 25 plants around the world that use that technology. But sometimes we get involved with recovery of catalysts, which are very expensive and can't be disposed by liquid-liquid extraction. And then again, using water to extract material from these things. In the last several years, we've done a lot of work in the biochemical processing, for instance, where chemicals are being produced by fermentation broths or algae broths. And again, if these things are higher boilers, it's not economical to try to boil the broth away. Those could be recovered by liquid-liquid extraction more effectively. Effluent treatment, certainly if you have a wastewater with high boilers or non-volatiles in it, liquid-liquid extraction is an excellent technology to consider for those types of applications. So this slide here is showing what liquid-liquid extraction is in its basic form, and just to give you some flavor for the terminology we use. In extraction, you have a feed that you want to bring in contact with an immiscible solvent, and you have something in the feed that you want to remove, and we're showing this as component C here. That we would call a solute in an extraction process. So you want to bring the feed and solvent together, mix them very intimately, and affect a mass transfer of that solute from one liquid phase to the other. So for instance, if this feed is aqueous, the solvent can be toluene, benzene, ethyl acetate, MIBK, any organic solvent that is immiscible in water. Then you want to separate the 2 phases. When you separate it, you get an extract phase, which is the solvent that's picked up the solute. You get a raffinate phase, which is the original feed that's given up the solute. Now, assuming you mix long enough to reach steady state, this would be one theoretical stage of extraction. And when you look at that stage, you have what we call the fraction unextracted. That's simply how much did not extract in the single stage. You have your distribution coefficient, which is defined at steady state or equilibrium as the concentration of the solute in the extract divided by the concentration in the raffinate. And we're showing that to be about 8 for this example. So obviously for extraction, you want to find a solvent with as high a distribution coefficient as possible for the solute that you want to remove, and low as possible for the other things that are in your feed. For most liquid-liquid extraction processes, you should be determining and getting your distribution coefficient. And then you have your extraction factor. That's the distribution coefficient times the solvent to feed ratio. In this example, we're showing that to be about 4. Again, higher the extraction factor, the greater your extraction is going to be. At Koch Modular, we like to use a rule of thumb of a minimum extraction factor of 1.3. And I'm going to show you that in a subsequent slide where we get that from. So 80% extraction is obviously not good enough for most extraction processes. So if you're doing a batch process where you're mixing and decant, which we call cross-flow extraction, after the first mix and decant, you then have to take your raffinate, add solvent to it, mix it up again, decant it again, and do that as many times as necessary to get from the feed concentration to the raffinate concentration that you're looking for. Usually, though, a better process is to do this in a countercurrent fashion. Okay, countercurrent. So this shows the same 4 stages as I had in the cross-flow example. However, the solvent you see now comes into the 4th stage, and then the extract from the 4th stage becomes the solvent in the 3rd stage, and so on in a true countercurrent fashion. Now, if you run through the calculations with the same number of stages and the same solvent to feed ratio, you can get to pretty close to the same raffinate concentration using countercurrent versus the cross-flow method. But obviously now what you've done is you've significantly reduced the solvent usage. In this particular example, you're only using 25% as much solvent. And usually that solvent needs to be recovered downstream say by a distillation process, and that's where the bulk of your operating costs for an extraction process is going to come in. It's going to come in that downstream distillation column and not in the extraction step. So certainly the idea of an extraction column then is to have your two phases flow countercurrently and then to create theoretical stages in this column to create a more effective extraction. In this example, we're showing the feed coming in the top, and it's the heavy phase, and it fills the column, so it's what we call the continuous phase. The solvent's coming in the bottom, being broken up into droplets, which flow up through the continuous phase to the interface. So that's the dispersed phase in the process. And then the extract comes out the top and the raffinate comes out the bottom. So which phase should be continuous and which phase should be dispersed in an extraction column is a very good question. Brendan is going to be covering that later in this presentation. One of the nice things about liquid-liquid extraction is it's very easy to generate liquid-liquid equilibrium data. We like to use this sort of a setup, a round bottom reaction type flask. It's something you can buy in any glass, any laboratory catalog. Bottoms take off, jacketed so we can control the temperature. So basically you put your feed, your solvent in here at a specific ratio. You mix them up. We find for most applications, 2 minutes of high-speed mixing is sufficient to reach equilibrium. You then turn the agitator off, let the phases separate. Once they separate, you take a sample of each phase, give it to the analytical chemist, and you get the determination of the solute in each phase, which gives you your distribution coefficient. You would then take the raffinate that you separated, put it back in, put fresh solvent in again at the solvent to feed ratio, mix it up again, separate it again. And we like to do this 4 to 6 times to get from the feed concentration to the eventual raffinate concentration that you're looking for, because the distribution coefficient can change with the concentration of the solute. So you can definitely generate your equilibrium data, your distribution coefficient. Sometimes you can use this to screen solvents. But we have a third point here: perform qualitative observations. From our standpoint, it's just as important as the raw data that you have a qualitative observation of how these two phases interact. And specifically, when you turn the agitator off, how quickly do they separate? What happens at the interface? Do you get solids building up, which we call a rag layer? Do you have a two-phase region, which we would call an emulsion band, where the two phases do not separate instantaneously? These are all good things to put down, and it helps someone like us as we look to the future to decide what type of column is going to be the best column for this particular application. So what you're doing when you're doing these mixed decants is you're generating your equilibrium curve, this blue line here, Y being the concentration in the extract phase or the solvent phase, and X being the concentration in the feed or raffinate phase. So every time you mix and decant, you're doing a lower and lower point on this equilibrium curve. If your distribution coefficient is constant, you'll get a straight line. Once you have the equilibrium curve, then you can operate your extraction column and use to develop the operating line and use the McCabe-Thiele type method to step off the number of theoretical stages. So on this operating line, the upper point here is where the feed comes in and the extract goes out. The lower point down here is where the solvent comes in and the raffinate goes out. So like I said, you draw your line and step off the stages. And this is a simple example we had where we were extracting some material from a fermentation broth. It was actually a dicarboxylic acid. The red line represents the equilibrium curve, which we generated in our laboratory. So once we had that equilibrium curve, we wanted to decide if to get 98% solute extraction at a solvent to feed ratio, how many stages would be required. Well, the upper point here again is the feed concentration, which you know. The raffinate concentration, the extract concentration, you know as well by material balance. The other end here, the raffinate concentration, if 2% is left, you know that. You know the solvent coming in, and you can see we step off 6 theoretical stages at those operating conditions. You can also do your calculations using a Kremser equation. Here's the Kremser equation, which is very nice because you can put it into a spreadsheet and very quickly look at solvent to feed ratio versus the number of theoretical stages. However, it only works if you have a fairly constant distribution coefficient. If your distribution coefficient is changing too much, this might not work very well. And then this is the Kremser equation in graphical form. Here you see the fraction unextracted. There you see the number of theoretical stages. So let me give you a quick, simple example. If you were extracting phenol from wastewater using toluene as the solvent, the distribution coefficient is 2. And if you used a solvent to feed ratio of 1, that gives you an extraction factor of 2. So if you wanted to recover 99% of your phenol, which would give you 1% unextracted, you then go over to the 2 curve. These are curves of extraction factor. So that's about your extraction factor of 2 curve. And it says you need about 5.5 theoretical stages. So again, these are very useful tools for looking at solvent to feed ratio versus the number of theoretical stages. I mentioned before this this extraction factor of 1.3 as being a rule of thumb as a minimum. And you can kind of see why if you look at these curves, because if you go to higher extraction factors here, you can see these curves are going down into very high levels of extraction. However, as you go lower, they actually start to asymptote, and you even get to a point where you can't get high levels of extraction. So it's not an absolute, but we do find it to be a good rule of thumb. So I just want to show you quickly 2 typical extraction processes. Acetic acid is one of them, and the solvents that are typically used, ethyl acetate or MTBE. So your aqueous feed would come in the top of the extraction column with the acetic acid. The solvent would come in the bottom. As it flows up, it will extract the acetic acid. You then would bring that solvent over to this distillation column where you would distill it and eventually recycle it back to the extractor. At the other end of the column where the raffinate comes out, there may be a certain solubility of that solvent in the water. You're going to go to a steam stripper or just a regular stripper and strip that out and recover it, and again, recycle it back to the extractor. And when you look at a process like this, you can definitely see why it's important to reduce the solvent to feed ratio, because like I said before, the bulk of the operating cost is going to be in this solvent recovery column. So usually the most economical process is to put your stages into the extractor, minimize your solvent usage, and you'll have a more economical extraction process. I just want to mention there's another type of extraction process called fractional extraction. That's where you bring the feed into the middle of a column. You bring 2 immiscible solvents at 2 ends of the column. In this case, we're showing an aqueous alcohol coming in the top, and that's immiscible in a straight chain hydrocarbon like hexane. But as that alcohol flows down, it will preferentially extract the more polar components from that feed. And as this solvent flows up, it will extract the more non-polar components. And then you go to 2 separate distillation columns to create, in this case, 2 separate products. I'm going to switch gears a little bit right now, and I'm going to talk about different types of extraction equipment. There's many, many different types, and they each have a place where they can be most useful. We don't have time to go to an exhaustive description of each of these, but there are 3 basic types. You have your mixer settlers, which are either batch or continuous mixer settlers. You have centrifugal extractors, which are high-speed devices that force the phases to flow countercurrently. And then you have your column contactors, static columns, which are usually either packed or trayed, and then agitated columns, which would have either rotating agitators or reciprocating agitators. And then there's also pulse columns. Today, I'm just going to cover 3 of these. We're going to talk a little bit about packed columns, and then we'll talk about rotating internals and then reciprocating internals for extraction columns. So this one just shows a simple example of a packed extraction column. Typically, you're talking about using either random packing or structured packing when you talk about packing for liquid-liquid extraction. I will caution, though, in my experience and data I have seen, there really does not seem to be any good indication that there's any difference in efficiency between random packing and structured packing for liquid-liquid extraction. You can see here there is a difference in capacity. Obviously, the structured packing has basically twice the capacity of random packing. But the reason that there's not much difference in efficiency is because in liquid-liquid extraction, as opposed to distillation, you're not trying to form a film on the packing, okay? What you're trying to do is create droplets. Droplets create the most surface area for mass transfer in liquid-liquid extraction processes. In fact, if the disperse phase starts to coalesce on the internals, you're going to lose surface area, you're going to lose efficiency. So it really is very important to pick the right material construction for your internals. In an aqueous organic system, water will preferentially wet metal. So if you have water as the continuous phase, you're going to want metal internals. Organics will typically preferentially wet plastics. So if the organic is going to be the continuous phase, then you probably want plastic internals. I have a note here about that one issue with pack columns could be it's affected by changing in wetting characteristics. If something plates out on those internals over time and changes the wetting characteristics, that would obviously be detrimental towards efficiency, and you do have to be concerned about that. Poor efficiency due to back mixing. Back mixing is primarily when the continuous phase turns back on itself, and you want to minimize this as much as possible in an extraction column. However, as the diameter of a column goes up, the tendency for back mixing will typically go up as well. It's what you need to factor into your scale up when you're going to larger and larger diameters, how the height of your column is also going to go up. But it seems to be more pronounced in packed columns than certainly than agitated columns that I'm going to talk about. Packed columns are also not good for fouling surfaces. Obviously, for what I just talked about, it could change the wetting characteristics. They don't turn down too well. Maybe you're lucky to get down to 50% of capacity, but if you try to get too low, you're actually going to lose efficiency. And they typically are best for systems with low interfacial tension, not high interfacial tension. For instance, toluene water is a high interfacial tension system. Now I'm going to turn to the two types of agitated columns. First one I want to talk about is a Scheible column. In a Scheible column, you have a series of rotating turbines on a central shaft, and those turbine impellers are sandwiched between inner baffles and outer baffles. So what you're seeing here is one agitated stage in a Scheible column. The way this works is the liquids are drawn into the impeller, into the middle, and they're sent out through the inner baffles, and then you get a very interesting mixing pattern, you know, between the inner and outer baffles and the turbine impellers. This is what gives this a very high efficiency. And plus all this baffling helps significantly minimize back mixing. So this column has the highest efficiency of any extraction column based on data that's been published in the literature. So it really is best suited when you need a lot of theoretical stages. Another very important feature is it has a very high turn down to about 25% of design without affecting the efficiency. We don't highly recommend it for fouling systems or systems that tend to emulsify, although it will handle these to some degree, but you'll see in a minute, I'll talk about a car column. And I also point out here that the agitation speed of a commercial Scheible column is not very high, and these are not high-speed devices. Typically, the lower one being the bigger columns, and the higher one being the, a smaller diameter column. Sorry, go back there. Okay, these are the internals of two different Scheible columns, just to give you a feel. The one on the left, this was a large Scheible column. So in this particular case, all you're seeing is the inner baffles here and the turbine impellers sandwiched between them. The outer baffles would actually be welded to the inside wall of the vessel when you slip this internal cartridge in. The one on the right is a full cartridge. This has both the outer baffles and the inner baffles all together. And on smaller diameter columns, that's what we use. And then you can see this Teflon edge, so that as you drop this into the column, that's what will seal those outer baffles. Next, I want to talk about the car column. This is unique and different than any of the rotating type columns. What you have here is a plate stack that's reciprocating up and down. It has very good efficiency and the good turn down like the Scheible, but the key to the car column is the uniform shear mixing. With rotating internals, you have a non-uniform shear across the whole cross section, but with this column, you have a uniform shear. Therefore, you create a more uniform dispersed phase particle size, and for that reason, we have found this is the optimal column to use for systems that have slow phase separation, like low density difference, or they tend to emulsify very easily. If you have a fermentation broth or an algae broth, you're almost certainly going to want to put it in this type of a column. Because of the large open area and the fact that there's no dead zones, every part of the internals is moving, it will also handle suspended solids very well. We have in fact put up to a 40% slurry through a car column, and it's worked effectively. So this is a plate stack for, this was about a 1 meter diameter car column. Over here, you can see one of the perf plates. You can see very large holes, large open area, which gives it high capacity. Typically, they're spaced 50 millimeters apart, these perf plates, although we will vary that depending on the process. Then you have these white baffle plates. Those are donut-shaped plates with a Teflon edge. That's the highest diameter in the column, so if there's any wall contact, it will be that. And then you have a spider plate here, one at each end of the column and one in the middle, so that when you tie this whole thing together with these tie rods, that's what keeps this to be a rigid structure. This is data published in the literature, efficiency versus capacity, and you can see the Scheible column here has the highest efficiency based on this data published in the literature. Before I turn this over to Brendan, I just want to point out when we're ready in an early stage of an extraction process to give you an idea of what the column is going to look like and what it's going to cost, we can do that. The things we're going to be required are these. First of all, we need your liquid-liquid equilibrium data. We need your mixing and settling behavior when you generated that liquid-liquid equilibrium data. Then we need to know what your feed is going to be and what your required raffinate is going to be. And of course, the design capacity for your system. With all of these things, we can, number one, we can select what we feel will be the optimal extraction column for your process. We can then do a preliminary design of that column, diameter and height, get a budget cost for that extraction column, which then gives you an idea of what this is going to cost you, and you can make a determination if you want to keep moving ahead with the process. The next step in any development is going to be pilot testing, and Brendan's going to talk about that. But pilot plant testing is critical if you want a process performance guarantee. So like I said, I'm now going to turn this over to Brendan, and he's going to take you through a lot of that pilot plant testing that I just mentioned.
So this is Brendan Cross, and like Don said, I'm going to be focusing primarily on pilot plant testing and different types of chemical phenomena that can happen and take place within an extraction column. So it's really difficult to just design and immediately scale up to a commercial scale liquid-liquid extraction column. And this is because there's a lot of interesting things that can take place within an extraction column that are really difficult to predict or anticipate ahead of time using just kind of standard simulation software or other process design tools. Three of these that I like to point out are the tendency of a system to emulsify. If you mix those two liquid phases together, does an emulsion form? That's really difficult to predict ahead of time. It can be really problematic when running an extraction process. Also, the effects of impurities or solids on an extraction column, particularly from a mass transfer standpoint, can have a huge impact. Also, and Don touched on this a little bit earlier, the impact of coalescing and wetting characteristics, particularly of the dispersed phase, on the internals of the extraction technology can be really problematic and difficult to predict and anticipate ahead of time. So the best approach really is to perform a pilot test, testing out that extraction process. And if it's successful, scale up from the pilot plant data with some known correlations and equations for the particular type of extraction technology that was tested. So before I get into some of those different types of phenomena that I mentioned, this slide kind of shows 4 different ways an extraction column can operate, depending upon the relative gravity of the liquid phases and which is selected as dispersed and which is selected as continuous. You can see in the upper right diagram, the solvent has a lighter density than the feed, and the feed's dispersed, so the feed is flowing down, forming the interface in the bottom of the column, and the solvent's a continuous phase and flowing up through the column. This is something else Don touched on earlier. How do you determine which phase is dispersed and which phase is continuous? There's 4 factors that we like to look at when we're designing an extraction test or coming up with a preliminary design for an extraction column on how to select which is dispersed and which is continuous. The first of these is the most intuitive, I think, and that's just if you look at the relative volumetric flow rates of the feed and the solvent. So for static columns, like the packed column Don discussed earlier, you typically want to disperse the higher flowing phase. And the reason for that is that you're counting on that higher, relatively higher velocity and momentum associated with the higher flowing phase to be broken up on those fixed internals. For agitated columns, we like to do the opposite, because this proves to be to lead to more efficient operation of the extraction column and also allows them to run at a greater capacity. There, you're kind of counting on those rotating or reciprocating internals to break up the dispersed phase, so you can easily disperse the lower flowing phase and get really good efficiency that way. Something else we like to look at is the relative viscosity of each phase. And from a practical standpoint, what you really want to do here is you want to disperse the more viscous phase. You can imagine if you had a polymeric type solution in an extraction column and you tried to make it the continuous phase, it would put a lot of drag on those less viscous dispersed droplets and really limit the extraction column's operation from a capacity standpoint. So the best thing to do here is just to disperse the viscous phase. Something else that Don touched on earlier was surface wetting. And essentially the key takeaway here is that you want the continuous phase to preferentially wet the internals. And you can kind of see that in these drawings here, because if you do the opposite and you disperse the phase that preferentially wets the internals, you're going to lose a lot of interfacial area, as you can see here. And if you lose a lot of interfacial area, it's really going to affect the column's performance from a mass transfer perspective. Something else we also look at is something called the Marangoni effect. And really the takeaway here is that you want the mass transfer direction to go from the continuous phase to the dispersed phase. So the best efficiency from a Marangoni effect perspective is to disperse the solvent. This is also something else Don touched on earlier when he was talking about using a test apparatus to generate liquid-liquid equilibrium data. And these are 2 different types of chemical phenomena that can occur right at the interface. The first is something called the rag layer, and that's just where solids build up at the interface, and you can see a picture of that here. The second is something called an emulsion band, and essentially that's where the dispersed phase droplets take a long time to coalesce and come together and form a single liquid phase right at the interface. And there's a good picture of that here on the right. So there's a couple of different ways you can combat these phenomena from occurring in an extraction column, because they can become problematic if they're allowed to build up over time. One thing you can do is just simply reverse the phases. I've seen that really helps from an emulsion band standpoint. Also with emulsion bands, you can slow down the feed rates into the column or reduce the agitation speed. Oftentimes that'll reduce the growth of an emulsion band and allow it to kind of be maintained at an acceptable thickness. For rag layers, whenever we design a commercial scale extraction column, we'll actually put nozzles around where we expect the interface to be, so that if a rag layer does form, that material can be pumped out right at the interface, and those solids can be filtered out using an external filter. Something else we often see in liquid-liquid extraction columns is the issue of entrainment. And essentially, this is just where you have discrete droplets going out the wrong end of the column. As you can see in this first picture here, the feed is the heavy dispersed phase, but you can see some droplets are going out with a continuous extract phase. So there's a few different ways to combat this as well. You can put a expanded head at one end of the column where you're seeing entrainment. This just slows down the velocity of the continuous phase, allowing those dispersed droplets to fall out. The second thing you can do, and we often do this when we're running pilot tests, is that you can put a coalescing element directly in the column where you're seeing entrainment. So the discrete droplets go onto that coalescing element, they coalesce and come together and then go out the right end of the column. Something else you can actually do as well, and we see this quite often commercially, is that you can put an external coalescing vessel. That's kind of shown in the last picture on the right here. And that's just an external vessel with its own coalescing element, which does the same thing as just putting a coalescing element directly in the column. The advantage of doing it this way, though, is that if for whatever reason you need to temporarily bypass that coalescing vessel, that could be done commercially. So you could clean out that coalescing vessel and its coalescing element while still running the extraction column. So it kind of minimizes your downtime. Flooding is another phenomena that we see in extraction columns, and it's another reason where it's really critical to perform a pilot test before scaling up to a commercial scale design. And essentially, flooding just means the dispersed phase is being held up within the column, forming a second interface. And that's an inoperable condition because if it gets severe enough, not only are you really limited from a mass transfer perspective, you'll have eventually both phases going out both ends of the column. So there's a couple different ways this can happen. One way is that if you just increase the continuous phase flow rate so much that it prevents those droplets from leaving the column, and that's what's shown in this first diagram. The second, more intuitive way to think about this, I think, is just if you put too much energy and rotational speed into the column, you're going to create finer droplets, and eventually they'll get to the point where they're so small that so much drag is put on them by the continuous phase, which is running at the same rate, that they won't be able to leave and you'll flood the column. When we run a pilot test for an extraction process, this is something we always look to find, is what is the flood point of the extraction column, because interestingly enough, very close to the flood point is often where the extraction column runs at its most efficient point. And we also like to know just where that flood point is, how easy is it for the system to flood, and then if the system does flood, how can we get out of that condition? Is it just by slowing down the rotational speed in the extraction column? Is it by reducing the feed rates to the column, or is it to the point where you have to stop the agitation entirely and stop the feed rates entirely? So this is a point that when we perform a pilot test, we always like to know where that flood point is. So typically speaking, to scale up a commercial scale extraction column, you're going to look at data from the pilot test, what diameter you ran at, what the commercial scale feed rates are, the height of the agitated section in the pilot test, and either the rotational or the reciprocating speed of the extraction technology that was used. And then from that, using established correlations and equations for the technology that you tested, you can scale up to a commercial scale design. And before I go into an example of how we did this with an extraction pilot test, I'm just going to give everyone a quick overview on our pilot plant capabilities. So we have the capability to test car columns at our pilot plant in Houston with a plate stack as high as 20 feet. We can also test Scheible columns that contain up to 150 agitated stages. We also sometimes test packed extraction columns and also rotating disc extraction columns as well. Quite often, we'll also test the downstream solvent recovery, distillation steps, or distillation for downstream purification following an extraction test. We kind of have the very typical analytical capabilities such as GC, HPLC, and IC, among others. And if we can't do it in-house, there's other labs that we have really good relationships with in the Houston area that can perform the analysis that you're looking for, most likely. This slide shows a very typical extraction pilot test flow diagram. You can see we have mass meters on the feed and solvent lines going to the extraction column. We have heat exchangers on those lines so we can control the operating temperature of the extraction column. And in this instance, the feed's dispersed with the interface at the bottom, the raffinate's leaving the bottom of the column, and then the extract's coming out the top of the column. So Don, myself, and Tom Lightfoot, who's our pilot plant manager in Houston, we wrote an article for CEP magazine back in February of 2018 that really does a good job of how to design a liquid-liquid extraction pilot test with a couple of examples that show how we optimize a few different processes. If you're an AICHE member and you have access to CEP articles, this is a really good one to review and look at if you're interested, because it gives a really good overview on how to run and design an extraction test. That being said, I'm now going to discuss a particular extraction process that we tested. This was a process where our customer was looking to recover greater than 95% of carboxylic acids from a fermentation broth feed stream that had about 5% acids in it. Ethyl acetate was selected as a solvent for this process, but our customer found that it emulsified very easily, particularly when they were performing lab tests, and also when they tried running their own pilot test using an RDC column. They found that the operation of the column was really difficult just due to the system's tendency to emulsify. They weren't able to get the recovery they were looking for, and they also had to use a higher solvent to feed ratio than they anticipated. They had to use a value of 2, and they wanted to use less than that. So since the system had that tendency to emulsify, we knew right off the bat that a car column was going to be a good fit for this process due to its uniform shear mixing that Don discussed earlier. So this is what the pilot test setup looked like for this process. It's really similar to that previous slide I discussed. Here, though, you can see the ethyl acetate solvent is the dispersed phase, and it's flowing up through the column, forming an interface at the top, and the feed's a continuous phase. So the mass transfer direction here is going from the continuous to the dispersed phase. So we're taking advantage of that Marangoni effect for greater extraction efficiency. Now I'm going to go through a series of runs that show how we optimized this extraction process and was able to make the process and the pilot test a success. So for the first 3 runs, we ran the extraction test with a fixed plate stack height, essentially just the agitated section that's performing the mass transfer within the extraction column. This was also run at a fixed capacity and at ambient temperature with a fixed solvent to feed ratio. So you can see in run 1, we got pretty decent levels of extraction, in run 2 as well, but we did flood the column at 50 strokes per minute. Then we tried reducing the plate stack height in an effort to kind of slightly reduce the capital requirements for the commercial scale extraction column. But you can see here we didn't make spec, and we actually flooded the column at 40 strokes per minute. We tried going to 10 feet. We made spec here, but after this run, we decided to go back up to 12 feet because we wanted to reduce that solvent to feed ratio further. And you can see in run 7 and 8, we made spec at 2 different capacities at ambient conditions with a reduced solvent to feed ratio. We then made what proved to be the key parameter change for this extraction test. And that was just running at elevated temperature. So when we ran at an operating temperature of about 45 degrees Celsius, we blew away the spec in runs 10 and 11. So based on the successful pilot test, we scaled up to a commercial scale design based upon run 7 and 11. The reason we chose run 7 in addition to run 11 is because it gave our customer the most flexibility so they could run this extraction process both at elevated temperature for higher levels of extraction, but also at ambient temperature for sufficient levels of extraction. When it comes to scaling up a pilot car column, and this was run in a 1-inch diameter pilot car column, you can scale up to a commercial scale column 1-to-1 based upon the capacity. So since that pilot test was performed at 650 gallons per hour per foot squared, and we knew the commercial scale feed rate, and we optimized the solvent feed ratio, from that we were able to design the commercial column with a diameter of 22 inches, so it would run at that same capacity of 650 gallons per hour per foot squared. Then we have a couple different equations that we use to scale up both the plate stack height and the commercial operating speed for the commercial column. So you can see the plate stack height worked out to be about 38 feet, and the anticipated commercial operating speed would be 32 strokes per minute, but that's something that can be fine-tuned when you're actually running the process in the field. And this is what the commercial scale car column looked like for this process. You can see it has a diameter of 22 inches, as I mentioned before. The plate stack height was 38 feet. We put expanded ends on this column, as we typically do with car columns, just to minimize entrainment. And the overall height of this column is about 15.5 feet. And you can see in the picture on the left, this extraction column being installed at the site in one of our process modules. And not only was the extraction column and its associated instrumentation controls and piping provided within a process module, we also designed a complete modular system that included solvent recovery. And on this slide, you can see that. You can see in the picture on the left module contains the extraction columns. And I can use my cursor to kind of show that here. And the module on the right has a solvent recovery column here. And then this is a small raffinate stripping column here. So we were able to develop and design a complete extraction system with solvent recovery based upon that successful pilot test. We also have a video that's up on YouTube that shows the performance of a car column. This is a really good video to look at and watch if you're interested, because it discusses and goes through a lot of the different phenomena that I discussed earlier, like flooding, different wetting characteristics, emulsion bands, etc. So if you're interested in watching this video, you can contact us. You can contact me through LinkedIn, and I'll send you a link to it. Or if you just do a simple Google search for car column YouTube, it should come right up, and it's definitely worth a watch. So with that being said, that kind of wraps up our webinar, at least from what Don and I are going to present today. As a company, Koch Modular supplied over 300 commercial scale extraction columns. We've also supplied hundreds of pilot scale extraction columns, and within our pilot plant itself, we've tested more than 200 different processes. So I think at this point, I'm going to open this webinar up to any questions that may have come through.
Great, thank you, Brendan and Don. Appreciate you taking the time to do the presentation for us. We do have a series of questions. I'll see how many we can get through before our time is up. Let me just see what we have here. I'll read one of the first questions, and then we'll see if Brendan, Don, you guys decide who wants to take it. The first question is, what is the effect of pressure on the distribution coefficient?
Well, I'll take that one. In our experience, it doesn't have any effect on the distribution coefficient. I haven't seen a case where it's had any significant effect on the distribution coefficient. And by the way, temperature also doesn't tend to have much effect on the distribution coefficient for most cases, but I have seen some instances where it does. But I would say that's kind of a rare instance as well. The big difference when running under higher temperature is your mass transfer rate's better.
Great, thank you, thank you. Another question would be, is there any rule of thumb on density difference for choosing between types of columns?
Well, I can take this one. For really low density differences, the car column's a better fit just because it has that uniform and more gentle types of agitation associated with it. For greater density differences, the Scheible column's a better fit, as long as the phases separate relatively easily, because you're just going to get more efficiency and more theoretical stages in a shorter agitated section height with the Scheible column.
Great, thank you. Another one is population balance considered in your sizing calculation in order to correct dispersed phase velocity?
Population balance being I guess the dispersed phase population balance. I guess that would have to be what I'd assume they're talking about in that particular question. I mean, for most extraction processes, we find the amount of dispersed phase that's actually in the column is usually between, say, 15 and 30%. I would say for high density difference, you would be toward the lower end of that unless you really can agitate it pretty significantly to create very small droplets to increase, the holdup of those droplets. I mean, obviously what you want to do is you want the two phases to flow plug flow counter currently. So you want to create as small a droplet as you can and have as much of that dispersed phase in the column as you can, because if you do that, you're going to get the best efficiency. And that's, again, why we run pilot tests to determine where those conditions will be.
And just to kind of add on that, this is something we really look at when we're pilot testing. We try to push the column as hard as we can so we get really fine dispersed droplets to maximize the mass transfer efficiency. And since we run all these pilot tests, for the vast majority of them in glass columns, you can really see what's going on inside the extraction column. So it's really another great benefit of running a pilot test. You can really just kind of see what's going on inside the extraction column and how fine of a dispersion that you're actually creating.
Great, thank you. Another one here is, let's see, is regarding recovered solvent pH, and can it change with time?
The question was recovered solvent pH, and can it change
Correct.
with time?
Correct.
Well, pH can certainly have an impact on the extraction process itself. If the pH in the solvent, these things, since these are typically operated continuously, it's possible, I suppose, that it could change over time. But I would think that'd be correlated to a change in the solvent or feed materials going into the extraction column. The only other possibility that I could think of is there is some type of materials degradation due to the materials used within the extraction column that could be affecting the pH of the solvent leaving.
Yeah, usually once you reach steady state in the column, it all lines out to a specific pH coming out the bottom, and it's not going to change. It just depends on how long it takes the column to reach steady state.
Thank you. Here's one. How can we find equilibrium data if it's not available in literature?
Well, if it's not, you can use simulation tools like Aspen to generate equilibrium data, but the best thing really to do is actually perform those liquid-liquid equilibrium tests that Don discussed earlier, because then you're using the actual materials that you're going to be developing an extraction process around, and that'll give you the most accurate distribution coefficients and possibly other things like separation factors. Because trace impurities can really have an impact on an extraction process. And if you just use something like Aspen and design an extraction column based off equilibrium data determined by Aspen, you may be missing a few things. Typically, the extraction processes that we deal with aren't really clean and just a few components. There's always other stuff in there that can have an impact.
Thank you. Another one came in and says that, does the azeotrope limitation for distillation apply for extraction process also?
Azeotrope.
Well, extraction is often used for systems that have that limitation where distillation is looked at initially, and it forms an azeotropic composition, say with a high amount of water. That's a lot of times where extraction can come into play and can separate those materials.
Yeah, a lot of times when you use organic solvents like toluene or benzene or whatever, it forms an azeotrope of water. But that's when you go to the stripping column after the extractor and you're stripping out your toluene from the water. It's going to go to an overhead decanter, and the water is going to recycle back to the stripper, and you're going to pull off the toluene and recycle it back to the extraction column.
Kind of essentially like that first flow diagram you went through before, right, Don? Kind of analogous system there. We use a common decanter.
Right.
Great, thank you. Another question is, what is the typical diameter for a pilot test?
I'll take one, Don. So for a car column, very typical diameter is 1 inch, and we scale up to commercial scale extraction columns from a diameter that's small without issue. We've done that for years, and we have a lot of confidence running in a 1 inch diameter car column. For a Scheible column, the typical column diameter that we use is 3 inch.
Yeah, I'll just say for packed columns, for structured packing, we typically would use a 3-inch diameter column. If you wanted to test random packing, you have to maintain a ratio of 1:8, the packing diameter to the column diameter. So depending on the size of the packing you choose and can get, you have to multiply that times 8. And for that reason, using a random packing, it means you're going to need a big column and do use a lot of material to do that type of a test.
Thank you. Another one came in. It says, could you tell us something about reactive extraction?
We've done some reactive extractions. Usually, it reacts at a certain point in the column, and when it reacts, it then becomes, say, the material then becomes soluble in the solvent, and the extraction occurs after the reaction step phase. If you look like at metals extraction, those are oftentimes reaction extractions where they have to convert the metals to a certain form first, and then use several stages after that to complete the extraction. So we've had some experience with that. Obviously, if there's exotherms, you have to be able to handle that, again, pilot testing is perfect though when you got a reaction extraction, because if you can get it to work in the pilot plant, then we're very confident to take that and scale it up to the commercial size.
Mm-hmm.
Great, thank you. Another one came in. It says, for packed columns, how can number of stages be translated into height?
By pilot testing. As far as I know that Coke Glitch, we work with Coke Glitch, and they do have, a program for taking liquid equilibrium data and for, interfacial tension and some other variables, viscosity, whatever they need to know. They plug it in, and they can, from that, kind of get an idea of the efficiency of their structured packing. However, they don't give a performance guarantee. So again, it comes back to, and when I've done this I let them tell me essentially what they think, but then I run a pilot test and get confirmation to make sure we're getting the height efficiency that will work for the process.
Mm-hmm. And then you can scale up to the commercial scale from there.
Right.
You always want to really to determine what that efficiency is, regardless of the technology that you're using.
Excellent. Thank you. Thank you both, Don and Brendan, for answering these questions. We do have more questions. Unfortunately, we're running out of time. I do want to thank everyone on the call for putting these questions forward. These were really good questions. So I hope that everyone has learned a lot around design consideration and potential applications for liquid extraction. If we did not get to your question, we will be responding to you personally with answers to these questions. And then there's also going to be a recording available of this webinar for you to access as well. Also, please be aware, as I mentioned, we will be having a follow-up webinar in a month from now talking about the benefits of truckable modules. So that'll be, stay tuned for that in the next upcoming weeks. So again, thank you all for joining. And if you have any questions or looking to have more in-depth discussions around particular problems or applications that you may have, feel free to reach out to myself, Don, or Brendan to schedule something afterwards. Thank you. Take care.
Thank you all.